Thank you very much, and thanks for being here. I know I'm in between you and lunch. I will try to make sure that I'm on time and speak slowly, as Susan just told me to do so. So let's see. 20 years ago, I used to introduce myself to people as a computer scientist, but today I call myself a microbial ecologist, and I can get away with that because I publish in ISME, and there are some requirements I seem to fit in now. With my group at the University of Chicago, we're interested in a bunch of different aspects of microbial life. And we study microbial diversity and ecosystem functioning. We usually do this through genomes or metagenomics, and I hope these terms will make more sense as I go along with my talk. Metagenomics is what we do to characterize the entire DNA content of a given environment by basically extracting all DNA, sequencing it with our poor chemistry that produces very tiny short reads. It makes it difficult to understand the larger genomic context. And then what we do, we assemble them, stitch them together. Perhaps you are familiar with the de Bruijn graphs and how people assemble these very complex information. Then we try to identify which larger contexts go along together that are coming from the same microbial genome. This enables us to study microbial life in these environments without having to cultivate them first. So it's very effective, very powerful, and both you heard from Lipping and Janet this morning. And we're also interested in understanding determinants of microbial fitness. We see phenotypes. We know microbes are responsible for some of them to a certain degree, but we always want to understand what are the genetic basis of these phenotypes so we can actually come to an understanding of mechanistic underpinnings of these ecosystems we're studying. And our group is using pangenomics, metagenomics, and phylogenomics in combination to achieve that in different systems. We're also interested in micropopulation genetics and evolution, just like everyone else in our field. And we do this through environmental omics and biochemistry. We recently focused on this aspect of microbial life with a bunch of colleagues of mine, and I'll talk about it a little bit. And recently, we started to be very interested in microbial physiology and translational dynamics. And we do this through meta-epitranscriptomics by sequencing tRNA transcripts from environmental microbes and try to understand how chemical modifications on these tRNAs regulate protein synthesis and ribosomal machinery, which is very interesting all by itself. But when I was invited to give a talk here, I thought about which one of these themes in our research would be most relevant to people who are interested in math problems and complex data science problems. And I decided to choose this one. And the rest of the talk will be focusing mostly on this particular topic. But before I continue, I want to thank all my colleagues who are here, members of my lab. This is all done thanks to or despite them. And I'm getting to talk to you here, but they are the ones who are doing the actual science. So just to make sure that it's mentioned at least once. So the story I'm going to tell you about has started with this very basic question: how does the genomic heterogeneity emerge within environmental microbial population? So, the analogy to this, I will use an ocean system and imagine a sterile ocean, and you just drop one microbial cell in it. And what happens in oceans, in world's oceans, is that there's this conveyor belt. A body of water that is in front of you will be in front of you in a couple of hundred years again, because things are mixed and there's a large-scale current systems that go around in a very predictable fashion, and then the water comes back in front of you. So imagine a sterile ocean, and then you're standing in front of it and dropping one microbial cell. Millions and millions of generations after, you will get back that body of water in front of you. And of course, this cell is going to be now many, many cells. And the question is, are you going to see, if you were to look at every single genome in this agglomerate of cells, would they be identical to the genome that you dropped in? Of course, as you can see, this is how speciation works. You know the fact that they are not going to be identical because polymerase makes mistakes. And that's why we're all here. We managed to evolve out of that ocean a long time ago thanks to the mistakes of polymerase. But then there are evolutionary processes that select for certain variants and then purify out certain others, etc. So this is a very interesting question, I think, within itself. Where is that heterogeneity comes from? And can we understand the determinants, the processes that influence the maintenance of that kind of genetic heterogeneity in micropopulations at very, very highly resolved levels? And then how does it drive the partitioning of ecological niches? And this is a very important question, especially for oceanographers, because people had been trying to understand why do we see Pacific Ocean and Atlantic Ocean look different from each other when you look at the microbial compositions. What are the processes that maintain that kind of difference? And of course, the first thing that comes to my mind is that selection, some adaptive processes are selecting for certain organisms in certain places. But there was this very influential paper, in our opinion, that suggested biogeographic patterns in ocean that we see. Not every ocean regime is identical to other parts, but there are local clustering of microbial communities and members in those environments. Hallwegger et al. Suggested that these patterns in ocean could emerge in a neutral agent-based model. And basically what they've done, they started with a very small number of genomes and they did a simulation with expected number of variants that will be generated every generation. And then they did not apply any selection, but they only studied neutral processes by looking at how the conveyor belt would go around. And as you can imagine, as these generations of microbes grow and change over the route, they leave behind a smear of population signatures that make locales look much more like each other. And they propose that that alone could lead to these partitioning of the ocean. So basically, if you think even more about this from a philosophical point of view, you realize that those neutral processes could lead to speciation. So maybe we're all result of some neutral processes one after another. So that's a very interesting thought experiment, and a very similar one to the one proposed by Hubble. If you know the neutral theory of Hubble about biodiversity. So it's a different kind of null hypothesis for perhaps oceans. But in practice, it is a remarkable challenge to study this question, to understand to what extent evolutionary processes, distinct evolutionary processes shape the biogeography and heterogeneity within individual populations in nature. Because there are three major problems we thought, and one of them is, first, you need to find someone, a microbial population that's everywhere, almost omnipresent. And when I say microbial population, I don't mean microbial species or even strains. I'm talking about something extremely close-rated, way beyond those boundaries of biology that have been defined based on some arbitrary metrics. And I'll try to clarify that a little bit going forward. And the second problem is you need a lot of data. Data to be able to detect and characterize the genomic heterogeneity of that taxon, let's say, for now, in many different environments. So in the metagenomic lingo, it's the coverage. So if you find a given microbe in certain abundance, it is more likely you will be able to ask the question, does all the genomes that belong to this particular population in this environment identical to each other or not? So we need abundance of data. And if we are talking about very complex environments, we need a way to be able to accurately characterize non-synonymous variation. So I'll get to all these three. We recently put this study out after 4 years of work with tremendous amount of contributions from Tom Dalmont, who was a postdoc in the group, and now he's a CNRS scientist in France, and Evan Kiefel, who is a grad student still with my group. And so this is a published study at this point. It surprised all of us, to be honest, to see it published after 4 years. This study is about SAR11, which addresses the first problem. This guy you see here is the most abundant microbial taxon on Earth when you think about numbers. Just to give you a brief idea about how abundant it is. There are 1 million to 10 million bacterial cells in a drop of seawater. When you go to surface ocean, take 1 milliliter of water, there are about 1 to 10 million bacterial cells. And SAR11 makes 30% of oceans, up to 30% of surface oceans are made of SAR11, this very interesting alpha protobacterial taxon, a heterotroph. And when you think about the numbers, extrapolate one drop of surface ocean to the entire surface oceans, and then imagine 30% of that is made up by a single taxon. It's kind of crazy. So the effective population size and the number of cells in the environment and the replication rate, when you put all these things together, this is exactly where evolution is happening. So a lot of room for asking the questions that we had been talking about. So we realized that what we are looking for is going to be within SAR11 group. But now we want to go into a level where we can discuss populations rather than entire taxa. So what we did first was to put together a SAR11 pangenome with available isolates. It has been extremely difficult to cultivate SAR11, but thanks to tremendous efforts from Steve Giovannoni, Mike Rappe, SAR11 genomes have been cultivated, because first they observed in marker gene sequencing datasets these very abundant organisms living in the surface ocean from amplicon studies, and they were like, what are these? And it all came down from there, and now we have these cultivars. Much more is coming from other groups, and yet here we have only circular genomes, 21 of them, and this, what you're seeing here is a pangenome. And basically, pangenomics is a way to characterize the entire gene content of a bunch of closely related genomes to have an idea about how gene distribution patterns look like when you look at the entirety of this picture. And here, every concentric layer is a genome, and every spork here is a gene cluster. And if there's signal, it means that the given gene cluster has homologous genes in all these genomes. For instance, here, when you look at this is the core part. Let's say this is the core genome of SAR11. So although they are coming from different clades, as people defined, these share all these gene clusters. And then there are singletons, for instance. Here there are, let's say, 500 of them belonging to this genome. There are 300 of them belonging to the other genome. So those are the genes that are not occurring in any other genome. Perhaps they are phage-associated genes, or who knows what they are doing. And there are genes that are shared within some groups, etc. So when you look at this pangenome, nothing sticks out. It seems all these genomes are relatively comparable to each other. There are some significant differences that also matches to what people have identified in the literature for these genomes, but none of them is sticking out as a particularly good genome to study environments. Then next question is how abundant each of these genomes in the environment. Can we maybe find a better candidate for what we want to do? And for that, we have used these Tara Ocean project metagenomes. For those of you who are not familiar, this Tara Oceans project is a consortium project, in fact, with many involvement of many institutions, mostly in Europe. And they basically jump into this nice yacht and go around and collect samples and sequence the crap out of ocean. And thanks to their efforts, we have these crazy large metagenomes coming from the surface ocean, from those stations I just showed you here. So from each of these locations, we have metagenomes with about 200 million reads. So now we can actually ask the question, among those genomes that belong to SAR11 that we know to be the most abundant taxon in surface oceans, is there a particular genome that's almost omnipresent and very abundant, so we can start doing some micropopulation genetics? And so basically what we did, we took this pangenome and then dipped into those metagenomes and wrote back to understand the read recruitment and therefore abundance of these individual genomes. And here the heat map now appeared on the right side, shows you how abundant they are. In a given sample. So each, just to orient you a little bit, if you remember this map, so for instance, this particular sample corresponds to that row up there, and this one is down here, and this one is somewhere in between, etc. So this basically shows us the coverage of individual genomes in individual samples. So one thing that emerges from this is, in fact, there are so many things to talk about this. This is a summary of 31 billion sequencing metagenomic sequences. So indeed, there are many things we can say about that. But I will just very briefly point out that in this group, 1A3, this particular clade that is also supported by gene distribution patterns that are all together, they're almost identical at 6NS level. They are very, very similar to each other. Yet you can see that there are some very distinct patterns, ecological patterns, you can observe from here. For instance, just to orient you a little more to this, because this is the important part, this particular clade is known to be associated with colder water temperatures. So, ecological insights that emerge from metagenomic read recruitment also shows that these guys are occurring in a bipolar fashion. So they are in those samples in Tara Ocean project closer to north and south poles. And then you have all these temperate guys, and that is also known. These are this is the clade with some cultured genomes that like warmer waters, and you see that the signal is mostly coming from temperate oceans. If you think about in terms of latitude. And there is this one guy here that recruits reads from, yes?
sorry, finish your sentence.
Oh, go ahead, please.
No, you go.
So there's this one guy that recruits reads from every single metagenome and occurring in every temperate ocean sample in high abundances. So this indicates that. So we in fact managed to find two things together. One, a genome that is almost omnipresent, so occurring in a geographically very distinct areas of Earth, and then recruiting tremendous amount of reads from metagenomes so we can do actually some population genetic studies.
Okay, can I go now?
Yep.
All right. So if you were to integrate, just so that I'm sure that I'm understanding your plot correctly, if you were to integrate all of the sequences along your one row,
Mm-hmm.
you basically would get back your part of your metagenome, right?
Mm-hmm. Yes.
So how much are these 21 genomes representing of your SAAV?
Great question. It's 5 to 15%, let's say.
That's pretty impressive with 21 genomes.
Yeah. What is perhaps even more impressive is that this genome is
Right, so
just so much more abundant than everything else around it, even though there are no particular genes that would indicate that this should be so different from everyone else. So as you can see, I know this this does not maybe immediately communicate the fact that this genome is not necessarily very, very different than these other genomes. Yet this genome recruits tremendous amount of reads, 400 million reads per metagenome, almost. So it's particularly abundant. We still don't know why it's the case, and that's a very important question for multiple people they are going after. But that's the case. So the take-home message from this figure for now is that we found a genome that is recruiting tremendous amount of reads.
I'm sorry, but you're not single nucleotide variants are being bin together, right?
Yes. So we will get there,
Okay.
because that's the entire purpose, right?
Sure.
So just to establish this point a little more, now we have an isolate genome, the name of which is HIMB83. It's a member of a SAR11 population in the environment, which we will call 1A35, because we divided 1A3 clade into multiple different clades. It doesn't matter what it is, but it dominates this particular population, which contains this as a member of it, dominates temperate surface ocean metagenomes.
what does it eat? Sorry, what does it grow?
I don't know. It doesn't produce anything. It's a bottom feeder. It basically eats what others don't eat. So it's a heterotroph and a slow-growing organism, but I'm not exactly sure what the.
Can I ask you just one more question?
Yes, sure.
Okay, sorry. Can you tell about phages and how are the difference between your genomes accounted from phage presence? Or
Yeah, usually what happens is it's very difficult to understand association between different genomes based on phages, because phages usually end up in these parts of these pangenomes. They have genes that are very unique to each prophage, so they don't add up to anything that would look like this, so start organizing genomes. So we find phages in these genomes, but we don't necessarily characterize these genomes based on phage information very much. Okay, so going back to the question about single nucleotide variants. So we're recruiting all these short reads from metagenomes. What this means is we go to a given metagenome that includes pieces of DNA from every organism that's living in an environment, and we take a genome and we ask the question, can we get every single short read that's nicely matching to this genome? But that nicely matching does not necessarily mean matching 100% to the genome itself. So it may be the case in a weird world, every single read that matches to our genome would be 100% identical to it, right? In that case, if I look at the frequency of short reads and their percent match to the genome, I would see a single peak at 100% and nothing else, right? But if there is heterogeneity in the environment, I would see a spread. And for instance, this shows you the spread from one metagenome. So basically, a relatively small number of reads actually are matching 100% to our genome, and the rest have this tremendous spread. And if you were to be familiar with the microbiology and what's going on today, there is this emerging understanding that 95% sequence identity at this level defines species boundaries. And SAR11 looks at that and then laughs, because it definitely does not work for SAR11. So it's basically Sarlevin is an oddball in that case, and this is not only recognized by us, but there are many other researchers who highlighted that. So this is the amount of spread you see, and in fact, you may have been able to see more if the mappability of reads did not slowly decrease due to bioinformatics artifacts. And when you look at the entirety of metagenomes, you realize that this single genome and the population it represents in the environment translates to tremendous amount of heterogeneity. That's what we learn from this. So we learn that these reads that are mapping to this genome is not necessarily mapping at 100% level. Does that make sense? Yes?
Are we supposed to take that as nucleotide diversity, or is there mapping error in that?
Nucleotide diversity. There is.
How do you know that you don't have mapping error?
Because we are very stringent with how we keep reads in BAM files. We expect them to map completely. We expect them to correspond to some quality standards that would tell us if there are insertions or deletions, or if they are truncated, they wouldn't be included in our analysis. And then I will tell you just a little more about how we make sure that we are not suffering from non-specific read recruitment coming from neighboring populations. So we are extremely careful about it because it's a huge, huge problem. In fact, that is the driving factor of many microbial population genetic studies. Non-specific read recruitment-driven variants creating an understanding as if there is a variance in the genomic backbone when it's just ecology. I know this is a lot to look at, but when you look at the so one thing I want to tell you is, now this communicates to us that there's a lot of heterogeneity, but this could be basically due to completely random processes, right? It could be, it may have nothing to do with anything non-neutral. So, but when you look at the statistical properties of these curves, you realize that there is some sort of association with temperature. It's not extremely strong, but it is there. So the skewness and the mean of where reads start to reach their peak for each metagenome, it seems there's a temperature component to it. So just to keep in mind, this indicates that this is not like a case of quasi-species where just things are mutating randomly. It does not necessarily tell us there is selection at this point, but it does tell us that it's not just random. So we will go through what this is. But if those of you who are careful maybe realized already that this is only talking about percent identity of metagenomic reads mapping to the core genes of this environmental population. Why? It's a very important question, and it kind of goes back to what Lipping was telling us. As a member of this environmental population, HIMB83, the genome itself, is not necessarily exhaustive to describe the entire gene content of the environment, right? It will have every gene the environmental core contains, but it will not have genes that are accessory to other members of this environmental population, if that makes sense. So just to give an idea about how it looks visually, this is how it looks like. So every spork in this case here is a gene from this genome, and every layer is a metagenome. And genes are organized not based on their synteny in the genome, but based on their distribution patterns. And you immediately see that there are some patterns, right? So those that are systematically found are those that represent the genomic backbone, and those that are found in some geographies and some, for instance, this is Indian Ocean, this is Pacific Ocean, and you can see that certain genes are more found in members of this population in those environments, while others are not found as frequently. So we are seeing a pangenome, but our job is to find out which genes can we rely on to do actual population genetics, because we don't want to be resolving geographical differences. We want to understand ancestral associations. Right? So just to simplify that a little more, if this is the genome, and when we look at metagenomic read recruitment results, and when we see something like this, basically what we want to do is to eliminate all these guys, so we are not dealing with presence-absence patterns as a function of geography, or some poor read recruitment because the contig has ended prematurely, etc. So we want to only focus on genes that are covered well in many, many metagenomes. For instance, like this one. And in fact, as you can imagine, you know, most of you know much better than I do, it's pretty straightforward when you look at the patterns I showed you in the previous figure. So when you go to this one, it is relatively easy to identify which genes are systematically occurring in metagenomes in a fashion that is also matching to other genes in the same metagenome, and those that are kind of missing systematically or randomly. So for instance, these are the genes that are missing in every metagenome but occurring in this genome. And that is another very important question to, in fact, discuss and understand as microbiologists. What leads to the emergence of these hypervariable genomic islands? These are real genes that are occurring in the environment, but in such small number of fraction of the environmental population contains them. And what maintains it is, I think, a very important question that will become more and more relevant as the years go by. So if we were to focus on a single gene, this is what we see. So this is a single gene from this genome, and these are the metagenomic read recruitment results. Our data kind of looks like this when we look at the variants. Reads that are not matching 100% to the genomic context. So there is a short read here, for instance, it is A instead of T, and then you account for a difference. And then if it's happening systematically, then you have a lot of variation that makes you feel like, yeah, I'm looking at something real. And I don't want to go into details, but for instance, if this bar is full, it means every single short read you are recruiting from the environment has a mismatch at this particular location of the genome. So from which you understand that the environment is in fact fixed completely. So the environment has no variation whatsoever. It just does not agree with your genome. But when you see something like this, you realize that, well, okay, right here, 50% of the reads have T, and the other 50% has C, which matches to my reference genome. So those start to become very interesting, right? So yes?
Well, wouldn't you think that if 100% of the sequences come out as T and your reference genome is an A, and your genome from the reference came from the environment, wouldn't you think that you had an error?
No, the short answer is no, but there are going to be, so this is a culture genome, this is a genome, it's a member of this population coming from a particular location in the ocean. Yeah. And cultivation is not necessarily giving us the most abundant organism, in even most abundant member of a given population, which is very hard to even think about. How could we even find out about that? But it is very likely that you will cultivate a genome that will have a mutation. Sure. In culture, perhaps, that will make it completely different from everything else. Even it will lose its connection to its own roots as population grows in culture, as it goes through various bottlenecks. Make sense? Okay. So when we look at these nucleotide variants, it becomes extremely difficult to make sense of non-synonymous variation because it's very complex. So we find often single nucleotide variants occurring more than once in a single codon. But synonymity, those variations in the genome that influences the amino acid sequence and likely protein function is a property of the codon, not the nucleotide, right? So we are used to, from eukaryotic world, that it's okay to study single nucleotide variants to single nucleotide polymorphisms to study synonymity versus non-synonymity, but in complex environment populations it does not work. So therefore we developed this idea that we could use perhaps single amino acid variants instead of single nucleotide variants. And single amino acid variants are like single nucleotide variants, but they are different because they focus on explaining the frequencies of varying amino acids instead of nucleotides. So at a given position, we do no longer count Cs versus Ts, but we look at isoleucine versus leucine, and it makes the data much more clean and enables us to ask some very specific questions I'll show you in a second. And the term single nucleotide variant, unless you have not heard it before, is a better version of the term SNP, which is much more frequently used. Because in the context of metagenomic read recruitment, when we don't know where our shorties are exactly coming from, we cannot talk about SNPs, because the term SNP requires you to be working with a single population. From human genome, if you're looking from human genome, you have SNPs, but a metagenome to a bacterial genome, you may not have SNPs, and you wouldn't know, so it's a less constrained term. And SAAVs, single amino acid variants, are better than SNPs if you're interested in accurate characterization of non-synonymous variation in complex populations. And just to give an example why it's the case, for instance, this is a gene, let's imagine, and it happens to have this codon here. And our metagenomic read recruitment looks like that when we're interested only in shorties that map to this codon. I hope it makes sense, because these DNA randomly chopped up in the environment, our sequencer gives us this. When you align and ask the question, show me all the reads that's mapping to this, you get something like this. And let's say you are looking at the single nucleotide variants and you saw this. A's and T's are occurring in this particular nucleotide position. And what's your conclusion? Is this synonymous or non-synonymous? Or to what extent this is non-synonymous? And A's are matching to A. Of course, these are synonymous, right? And then T's are not matching A. But do we have any TG in this set of codons that can decode serine? And we don't, we have one for, no, we don't have any. So, okay, we say this is probably 50% non-synonymous, 50% synonymous, right? And then we look at the pileup of the next, and now we see C's and G's. And G, we're fine, it's synonymous because it's matching to the nucleotide of the genome to which we're recruiting these B's. But C's are not matching. But do we have CT? And we have CT among the codons. So we think maybe this is just differential codon usage. So this is very, very likely synonymous. So we conclude this is 100% synonymous. And then here we see all the T's, so we're happy. So now, what we do, we discard all the reads that are partially mapping to this codon, and we take only those short reads with nucleotides that are physically connected to each other, right? And now we count only these codons to understand the variation, and look what happens then. This, in fact, two of them, they basically all of them encode for a different amino acid. So it's 100% non-synonymous. So, and you missed that. And the problem is, this is not a very extreme example. This happened 22% of our data when we were working with SAR11. And this was necessary to be able to make sense of this non-synonymous variation accurately. Okay, so what happens at the end of the day, you end up with a tremendous amount of data. So we had one genome that recruited 0.4 billion reads from our metagenomes, which result to 100,000 single amino acid variants. The first question you could ask is how these single amino acid variants, what do we find? What seems to be variable in the genome, in the environment, and how is it distributed in the genome? Because if, for instance, something is 1% of the genome in amino acid, and if it's variable in the environment 1% of the time, and if it's the same thing for something that occurs 20% and 20%, there would be a perfect linear relationship, then you could say variation is completely random, right? But when we look at the amino acids, how much they differ from an expectation if everything was were to be random, we see that, for instance, glycine is not found in single amino acid variant positions, but it's very frequently found in the genome. So you can see there are some discrepancies. When you look more closely, you realize that all positively charged or neutral amino acids are appearing here, mostly, except two of them, in fact, I'll mention. And others are, so there is some chemical association between how much they vary. And in fact, when you ask the question, is it about hydrophobicity of these amino acids, you realize that yes, in fact, the average solvent accessibility is really explaining the amount of variation. So those that are buried are not varying as much, which is expected. So complete random process does not explain this, but neutral evolution does. So purifying selection are removing those, fine. So we still are not close to having any idea about whether there are any adaptive processes happening or not. So the next thing you can ask, I'm going to take a single amino acid variant position, and I will look at the frequencies of amino acids there. And then I'll call them amino acid substitution types. So then when you ask the question, when I look at amino acid substitution types, what are the frequencies of those guys and their occurrence in different environments? This is what we see. So I'm sorry, these are all very complex figures and I'm flying through them, but I hope you will stop me and ask questions if you get confused. So basically what this figure shows us, let's focus on this one first. Isoleucine versus valine. So basically we are looking at single amino acid variant positions in which isoleucine competes with valine, okay? So when we look at how they compete with each other, like whether their distribution resolves to anything regarding temperature, for instance, we see a flat line. So there's nothing going on between those guys. So it's completely neutral. It does not care about the environmental signal, you may say. And then when we look at this one, though, we have an association with temperature. And then we have an association with temperature over there. So it shows that amidst the purifying selection, there seems to be some signal for environmental processes that seem to be influencing composition of amino acids and amino acid substitution types. But when you ask the question, can I explain it by looking at whether these are negative charge or hydrophobic amino acids, you don't see any association. Why? It is largely because we are just lumping together all sorts of information without paying attention to physical chemical properties of the protein context. So this is a very important point, I think, that we miss in population genetics study. Yes?
So at the ground level, is what you're hypothesizing that by changing this individual amino acid, the reason why they change with temperature is because now the interactions between the proteins may be better stabilized?
Exactly.
And do you have any structural data to come
Yes. So the next question, of course, is to bring in predicate tertiary structures to understand when we talk about isoleucine versus valine or any other pairing here, where do they occur? And it's a very difficult task because SAR11 is not necessarily a very popular organism among those who do X-ray crystallography. So we don't have a lot of understanding of tertiary structures of genes that are occurring in the genome. But what we did, thanks to Evan's help, we downloaded all the templates matching to our genes from PDB and did dynamical modeling, starting with those templates. And then we got all these structures for some of our core genes, which enabled us to ask all those variants we see, where they occur. For instance, I'll show you a couple of interesting examples. For instance, here we have this glutamic acid occurring at position 17 of this gene, and it's here. So there, glutamic acid versus valine is competing. And as temperature increases, the ratio between glutamic acid and valine changes. And in contrast, glutamic acid, so there, glutamic acid frequency is much higher in colder temperatures. You see those are the red dots going down. And here, glutamic acid shown in blue, and then it increases in its frequency as a function of increasing temperature in this particular position. So the same amino acid behaves differently, responding differently to the same temperature changes in different contexts. So this tells you that when you lump all this information together, you are missing potentially critical details of evolutionary processes. And another example to that is here, for instance, tyrosine in position 24. So the red one here is competing with another amino acid, and it decreases in its frequency as temperature goes higher. And when you look at tyrosine in here, it does not care at all in this position here. So, but how much can we look at these individual variants without really having some proper understanding, proper model of how to study these things? So it's very difficult, and we did not do that because we lack those insights and we lack those tools. And that's why I wanted to bring up the story here, because I think there's a lot to do here for those who can think in numbers. So just going back to the original question, so we see that there seems to be some signal that indicates that environmental parameters influences different parts of these proteins in different ways that causes these amino acids to substitute in different proportions. So can we make sense of it in a more comprehensive fashion? So we collaborated with two computer scientists, a very talented researcher, developed an automatic an auto-clustering output layer for a deep learning algorithm. Basically, we could ask the question, based on single amino acid variants across these metagenomes, which metagenomes look alike in the context of the single genome? Because now we're going to go all the way back to the original question and see how metagenomes relate to one another. So from estimated distances that emerged from the deep learning algorithm, we were able to organize our metagenomes in a fashion where metagenomes in which the SAR11 population had similar amino acid variants would come together. I guess that makes sense to a lot of you if you have been looking at population studies, how people organize human populations, etc. So it's not very different than that. But in this case, we're using single amino acid variants from the core genes of the environmental population. And as a result, that emerged two major branches. So the entire tree splits into two. And then, of course, what we could do is to see how they look like in the context of Tara Ocean data. And when you look at this, the first thing I had realized, and now I'm very embarrassed to mention, but I feel like it's my responsibility, Tom Dalmont and I, we were looking at it and we realized all the red dots that are coming from that branch, so somehow close to each other, is the eastern side of all larger continents. Look at this. And then all the blue dots are coming from the western side of really large continents. So it seemed to explain some interesting phenomena, and we were thinking about, well, I was largely thinking, I shouldn't blame him, high altitude winds maybe carrying over, influencing lower levels of dynamism of atmosphere and carrying over iron and other nutrients from whatever. And then Mike Lee, a colleague of mine, he was a PhD student at the time at the University of Southern California, he said, Meren, don't be ridiculous, these are large-scale ocean current temperatures. And I Googled it and I was not convinced. And then someone else, Ed DeLong, told me that that was the case. So then Tom and I Googled it again, and it was really matching much better to ocean current temperatures, large-scale ocean current temperatures, rather than the ridiculous hypothesis we had. So what seems to be happening is this current goes from here to here, whatever Lagrangian time passes. These populations are exposed to warmer and warmer temperatures. Because the population size is so large and they reproduce and they are exposed to new environments as they go along, they select for, perhaps this is one hypothesis, select for those amino acids, they reinvent those amino acids that will help to maintain the stability of their protein landscape to be able to fit to these warmer temperatures. And it goes like this and comes back up, and then they go back to cold variants. But the critical point is, if everything was neutral, as the null hypothesis posed, then we wouldn't have near identical single amino acid variant frequency profiles across large geographies. But when you look at this one of the best examples I have, two sides of Panama Channel is identical to Red Sea and Indian Ocean. And these are the most cold samples in our data set are matching to other most cold samples in our data set. And all over the place you see some associations, and when you divide them into prototypes, environmental prototypes, they become much more clear that that there is something that we cannot explain based on neutral processes alone. So the use of this is basically the highlight of the entire talk. If you didn't like it, then I'm sorry. But there is one more thing I want to mention before I wrap up, because I think I'm running out of time. The use of single amino acid variants also enables us to combine this information with protein biochemistry, at least by visualizing their distribution on these predicted contexts. For instance, what we see in the C, prototype C, is the coldest samples in our data set here, C, 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 C. And when you look at this, you realize that C, the same gene in C environments accumulated a lot of variants. And in warmer temperatures, it's much more, do not necessarily responding to temperature as much. This timberel protein accumulates variants only on the outside. The inside of it is very much fixed, because purifying selection makes sure whenever you tinker with that, you go out, you no longer function. And for instance, this is an example that I like a lot. This is a 3-domain protein. And by looking at this, you can see that there are these 2 domains you should never touch, because that is what the nature of this protein requires. Purifying selection will just take you down. So this is in fact a very critical information to those who are trying to improve protein function. You have an ethanol production system, and now you can take your genome, go to the environment, and ask the question to an environment that has been doing this experiment for millions of years to see which parts of your protein you're allowed to play with. And then, based on some perturbation experiments, you can select for those that will likely give you a competitive advantage if you were to rely on those variants, perhaps. Yes?
So you're probably right, but you're kind of eyeballing the data. Is there an energy calculation you can do on the proteins to really prove that they become more stable?
Excellent question. So there's one thing we did. We looked at the, I don't want to scoop Evan, but we looked at the association between sizes of amino acids and their placements in protein tertiary structures and temperature. And what we saw, the first thing that emerged, it seemed it's always the case that the smaller amino acids are being replaced with larger ones. So, and you go back 30 years, biochemistry tells us that in order to maintain protein function and stability in colder temperatures, you need to influence its rigidity by replacing smaller amino acids with larger ones. So we saw evidence of things, evidence in quotes, but we did observe certain things, but we're not biochemists. We're trying to get biochemists interested in these kinds of things. So environmental omics, biochemical insights could come together perhaps to develop better ideas. And oceanographers also. We are not oceanographers either. Yes?
In the previous talk, it was mentioned that short-chain fatty acids kind of create this distribution of acidity in like digestive pathways. Could you use a similar analysis to kind of understand the proteotypes based off of
Excellent question.
pH?
So you're saying that you could look at different pathways in these genomes and how they are influenced by these variants differentially perhaps to understand their relevance to the environmental requirements. Temperature, we focused on temperature because temperature is such a foundational perturbation, the entire genome responds to it. Every protein needs to do something about temperature, pretty much. So we did try to see whether there were clustering of some variants in certain genomic pathways, and even did observe some interesting things, but it never led to anywhere where we felt comfortable to publish. Yes, so I have to finish. And so I will skip that because between these two yellow lines, I am listing all the limitations of what we have done. So it is extremely important to take everything I said with a grain of salt, most of which can be tested with experiments. Some of those we will probably never understand fully, but since the paper is there, I'm not going to go through this. To summarize, single amino acid variants are promising tools to tease apart evolutionary forces acting upon naturally occurring micropopulations, and can reveal structural traits that are likely influencing fitness by linking biochemistry and organismal evolution. And this was a case where we were interested in an ocean microbe, but you can see that this could be applied to any system, any microbe, with the presence of metagenomes and genomes and good coverage. So with that, I thank my group and our funding sources and you for your time. And just a quick plug that everything you've seen here, including pangenomic analysis, metagenomic read recruitments, their characterization, single-amino acid variants, and then their mapping on tertiary structures and prediction of those things has been done in Amio. And this is a platform that we have been developing, which is 80,000 lines of code at this point, and a lot of online tutorials, more than 120,000 words now. And extremely flexible, that I think opens the door for those who are interested in implementing algorithms to work with data without having to think about how am I going to find metagenomic read recruitments and tertiary structures to combine these two to make predictions. So, and it comes with extensive visualization capabilities. All these things are screenshots from Amio visualizations, and they're all interactive. And Amio is developed largely by these three individuals beside me, and we are lucky to get help from a large community out there. So it's more of a community platform than our tool. So if you have any interest, we can talk more to see how we can support your interest to be able to implement algorithms for different omics approaches. So thank you very much.